Not really. It's so because we have IP. Maybe we can welcome Lenore, and then we can keep up with you. Keep going with your questions. Hi, Lenore.
Hello.
Hello.
So it's Thomas?
Yes.
Or Thomas?
Exactly. Nice to meet you.
You too.
Yes. So thanks for being here, Lenore. Thomas was just explaining to me how he came across our lab and the kind of internship he has to do for his CEGEP. So which is the course that you're taking?
It's projet intégré. Basically, the name is basically to find a project where we can have an internship. It's the new concept of Collégiale International Saint-Anne. That's basically what they're selling to everyone, and we have the choice to find a project in other countries or whatsoever. And basically, it's just on a specific subject, and then we find an internship where we try to push the researchers on it.
Nice. So coming back to your question, so we work with induced pluripotent stem cells, probably, right?
Yeah.
You heard about them? You know how it works, how to generate them?
Yeah.
Okay. So basically, because we take what we have here is to explain how it works at our institute. It's not just our lab, but it's also the hospital side that is involved. So we get samples from patients and from healthy subjects.
Mm-hmm.
So they collect the CBIG, which is our biobank, will collect blood and urine samples, any samples that can have somatic cells.
Mm-hmm.
And then these somatic cells will then, by our lab or even sometimes by other labs or by the CBIG, will be reprogrammed. So we'll push them, we'll give them factors to turn them back into stem cells. So these are the induced pluripotent stem cells. And then from that, we can do genome editing, like by CRISPR technology. We can do a series of manipulation, but also interestingly, from the iPSC, you can generate any cell from your body, right?
Mm-hmm.
In our case, we are mainly interested in the central nervous system, the cells of the nervous system. So neurons, microglia, astrocytes, and oligodendrocytes. And because we are generating these cells from the patient or from a subject, they will carry the same DNA material from that patient, from that subject. So we study, so going back to your questions, whether we study Alzheimer's or not, we study diseases that have a genetic component. So I think, Lenore, do we have some work in Alzheimer's now? I don't think so. No, we used to have it in the past.
More what, sir?
Some studies with Alzheimer's disease.
Not really. Our main focus is on Parkinson's disease and ALS and some neurodevelopmental diseases such as autism spectrum diseases.
Because for Alzheimer's, it's a small portion that it's genetic, right? Most of Alzheimer's is familial cases. Sorry, Lenore, I interrupted you.
Yeah, so I'm trying to think of an Alzheimer's specific project we have. I don't really think we have one.
I think at some point we're generating cell lines for Alzheimer's disease, but I don't think we use the cell lines. So one thing we do is that we can generate cell lines by CRISPR. So we can have, for example, we can take a patient line. So someone that has Parkinson, for example, that we have a known mutation. And by CRISPR, we can we can remove that mutation and then turn the cells into healthy cells. So this will be the healthy cell line that it's coming from the same person. So it has all the genetic background minus the mutation. So you can compare, sometimes it's easier to compare because it's not two different genetic backgrounds than if you were, for example, just comparing with a human subject, any other human subject. So, so that's one of the things we generate the cell lines, and then other groups can use it because we work through an open science concept.
Mhm.
So basically, everything that we develop in the lab is openly accessible to other researchers, to the general community, and that's how we work. So I don't know if it answered your question.
Yeah, absolutely. And I was wondering, do you touch chronic pain in your lab?
No, we don't have any pain models so far. I think we're starting to develop a bit the sensorial neurons, and I'm not too sure what's the path for that, but I think it's because, so like Lenore mentioned, we do study neurodevelopment disorders, and some neurodevelopment disorders will come with some changes in sensory too.
Mm-hmm. And when you're talking about neurodevelopment disorder, does it has to do with like the myelin sheath and the neurons, or you're more in the another aspect?
We don't really deal with multiple sclerosis either. I was just looking at our cell line catalog, and it seems to concentrate on the healthy controls, the Parkinson's disease, ALS, some CRISPR knockouts of various genes, which maybe other diseases, and then some other. Yeah, as I look at them, it's mainly Parkinson's and ALS.
Okay, and for the Parkinson's, could I have like a little bit more information on what's going on exactly in the brain that gives this this disease basically?
Do you have anything there, Lenore? I don't have a slide on Parkinson. So just going back to your question about neurodevelopmental disorders, this would be disorders that are happening when the fetus is developing in the belly of their mom, or sometimes they start a little bit later on, but during the critical phases of development. And what is important, what is the focus for us is diseases that have autism as one of the symptoms. So they are diseases that affect the brain, and so that's what we study.
And is it clinical? Do you have patients and do you work with kids?
We don't, but at the institute they do. So we are working with cell lines and organoids, so mini brains, things. I don't know if you have heard of them, because you can have 3D culture, so we call it mini brain. So you can have a brain on a dish. Basically, it's not a fully formed brain. It's more like in a developmental stage, like in early developmental stage, and it doesn't have all the complexity of the brain, but does have a 3D structure. But we don't work with patients. The patients are at the hospital. We don't do any clinical work.
The clinician, like the doctor and the team, will ask the patient permission, like the patients give consent to have their blood collected, and it's another team that collects the blood and stores it, and then we can get the samples to reprogram into the stem cells. And for a guide to Parkinson's disease, I just put a link in the chat.
Do you want to open it?
That's the website, and I think they would. You can download it.
Okay.
Like Thomas, if you.
Yeah.
You can see it.
We also have some videos in.
I was going to say it's specific neurons in the brain, in a specific part, the midbrain, that die over time during the development of Parkinson's disease. And this causes usually movement disorders, so the patients can't walk as well. They may have tremors in their hands, problems speaking, and over time this gets worse, and there's other, what's called non-motor symptoms, other symptoms that develop. But as Louisa was just saying, we just deal with the cells in a dish or in 2D or in 3D if we make brain organoids. And then we see how well they have the same changes as cells from a patient with Parkinson's disease. And if the cells die off, one of the papers that was published was these cells had a triplication, like more copies of a gene called synuclein. And in those, the organoids made for those cells, the neurons died off. And there were other changes. So I can just find the link to that paper.
Okay, so basically it's in the
model.
DNA, and by taking like the stem cells, I think, could you kind of replace them, the ones that has like a genetic disturbance? Could you replace them, or it's too small, or there's too many? How does it work exactly for any possible treatment or?
Well, we're not using them for treatment. Other researchers are looking at using them for our treatment, the stem cells, but we're just trying to understand how the cells with the genetic changes, what goes wrong with them.
Okay.
And how we can measure them by looking at, say, the proteins they make, how, have you heard about mitochondria?
Yeah.
And how parts of the cell don't work very well in cells with this genetic change.
Yeah, there is a promise for regenerative therapies, which what you're talking about, which is to use stem cells to replace disease cells and things like that. But there's still a lot of I think there is a lot of things to be figured out before it can really become a possibility, especially in the brain, because we understand so little of the brain that we can't just add cells there, and how we're gonna make sure that they make the proper connections, that they don't become a tumor. So there is a lot of stages still. And like Lenore said, that's not our approach. We use the stem cells more like to model the disease, to reproduce what happens in the disease, to find new things, to understand, these changes, the pathology, and also to look for possible medication too. So we do have part of the lab that works with the pharmaceutical industry, and they will send us compounds, so drugs, not drugs that are in the market yet, but that they would like us to test and see if they could become a drug accessible that is available for treatment. So we do have that part.
So we could test the cells in the dish and say, okay, if we add this chemical, will the cells, like with the genetic change, behave more like normal cells?
Yeah.
And when you do that with, say, 5,000 different chemicals, that's called drug screening. And we can do that with very powerful machines that can screen hundreds of different samples at the same time.
Okay. And have you tried these tests on the rats, or you're staying on the cells on the dishes?
So we don't do any animal work, but so if compounds get selected, but get picked up when we're testing, they will do, I will go back to the image that I was sharing here with you. It will go through the other phases. Let me just a minute.
So if you got this, the second link, that's to the article about the midbrain organoids.
Yeah, and I can send you videos too, Thomas. We do have videos that will explain the research that we do. That you might be interested in watching them too.
Yeah, that would be great as well.
Yeah. So yeah, so like you're hinting, there is the phase of preclinical model. So let's say we develop, a good model for Parkinson. Everything is working. We screen the compounds, like Eleanor was saying, all these chemicals. We look at them. We found one that it's really good. Then we would go through preclinical models, which is the animal studies. But we don't do that. So we basically do from reprogramming all the way to disease modeling in a dish. All the other steps are not done by us. So then from the preclinical models, then if they look at toxicity, is this toxic to the animals, does it kill the animals or not? They find the right dose that they would think that could work for humans, and then they will go for the clinical trials. But we also don't do that part.
Okay. And I was wondering for Parkinson, for example, it's a problem in the cells that affects afterwards, like the information that is getting transferred, which can give you like pulsation with your muscles and all. But could it affect the muscles that we don't actually control? For example, the heart or any muscle. I don't know how it's called in English, but it's muscle strié in French, I think.
Yes, it's striated muscle.
Yeah, could it affect those muscles as well, or it's completely different?
Well, there are some other muscles that are affected. That's like the non-motor symptoms. Sometimes problems with constipation, bowel.
Yeah.
So I don't know about heart problems.
Yeah. So the heart, I don't know either, but there is a theory that synuclein will spread like prion, so you wouldn't necessarily only see that in the brain, but like, the synuclein could spread like the prion disease does and will spread to other parts of the body. So it's not quite clear yet, but yeah, you could have... for sure, you have other organs that are affected. It's not just the brain.
Okay.
Just looking back on the symptoms. Bladder dysfunction, sleepiness, double vision, changes in taste and smell, insomnia, choking and swallowing difficulties, REM sleep behavior disorder. So there can be other symptoms associated with it. And not every patient has the same symptoms.
Yeah, and unfortunately the medication sometimes comes with its adverse effects too. So by using the treatment, they also start exhibiting some other symptoms that are related to medication. Memory impairments is one of them. The thing is that, with the progression of this disease, the degeneration, so the death of the neurons get more and more profound. So it gets more, the symptoms will get worse. And also, like Lenore was saying, the disturbance in REM sleep, it's something that a lot of people argue it's an early sign of Parkinson's disease. So you could actually watch, the sonogram, when you have the traces of the pattern of the brain activity while you're sleeping, and this would be very distinct than a person that it's healthy, and even before they start exhibiting the classical symptoms of Parkinson.
Mm-hmm. And is there anything you can do to push back like the Parkinson effects? Is there anything you could do like to postpone like the if what?
To delay?
Yeah, exactly.
To delay, you mean?
Yeah.
Currently, no, there's some medications that can be used to control it, but it seems like it's a progressive disease. And as Louisa was saying, after a while, the medication may not be effective. So it's unfortunately, unlike cancer, where we've made a lot of progress over the last, say, 20 years, there's been very little progress made in these neurodegenerative diseases. And that's why I would think that using human cells as models will help us make progress. Mm-hmm. And maybe give us candidate medications to test, first in say, the animal model and then and in clinical trials in humans.
So our approach is based in two different things. Like Lenore said, using human cells, it helps us, have a better model for testing, for understanding the disease and testing the medication. But also we work in this open science. We use the open science as a way to try to increase collaboration and just harvest different powers, from different groups and exchange information to really try to accelerate the discovery in neurodegenerative disease, because if everyone is closing their lab, just doing research and publishing in small groups and keeping the data for themselves, we might be doing the same work here that someone is doing in Japan, that someone is doing in Australia, and we're just, we're not advancing as much as if we were exchanging the knowledge, and we could be like building a puzzle, complementing each other instead of everyone trying to do the same work. So, like Lenore was saying, the problem also with the treatment is that you're treating the symptoms, right? That's why you can't really delay Parkinson's, because you're not treating the disease itself, like you're not preventing the neurodegeneration from happening. You're basically just replacing the neurotransmitter, which is dopamine, that is decreased, because as the cells die, the start dopaminergic neurons, which release dopamine, then you have dopamine will lower, you will decrease in the brain. So you want to replace the dopamine that it's being decreased, but you're not really preventing the neurons from dying. Mm-hmm. So it's kind of like a band-aid, like you're not really taking care of what's going on there. And basically it's also because, like Lenore said, we understand very little of the brain, so I'm not even quite sure why this is happening, Like we have an idea, but we don't have all the elements to be able to have a medication that could, like you said, delay or prevent, Mm-hmm. And because also by the time that we discover that Parkinson is diagnosed, the degeneration is already kind of severe, so you can't really rescue when it's, a huge percentage of the neurons already dying. So it's kind of funny in that way.
Yeah, and that's why I heard of the Michael J. Fox Foundation. Is it like Michael J. Fox has Parkinson's disease, so he's an actor. He was in Back to the Future.
Okay.
And he set up a foundation to provide funding for researchers and resources for people with Parkinson's disease. So I just, again, in the chat box, I put the links. And there's a lot of information there. And they're providing funding to try and find biomarkers for Parkinson's, so that a person, if they could detect it early, maybe there's a better chance of giving a medication that could prevent the loss of the neurons. And so they have a very large group where they're collecting samples and give money to researchers to look at it, to see what they can find, to sort of measure it. Because if you had a measure of how fast a disease is progressing, then you could say, okay, if we give the patients this drug, it helps slow it a bit. If this drug doesn't, and then you could do what's called the clinical trial and test out these drugs in, like if you've ever heard of FDA, the federal drug agencies, the states, that they would approve it, but they need to see that the clinical trials work and that they help people.
Okay, so right now we don't even know at what speed it spreads, and I guess it's different between everyone. So
it's also because by the time you start exhibiting the classical symptoms of Parkinson, the time that your doctor diagnosed you with Parkinson, the damage to your brain is already so severe that you can't really rescue. It's like, I don't know the exact number, but let's say it's like 70% of the neurons already degenerated. So like you have 30% left, like, it's a different thing. Like Lenore was saying, if you could have a marker that comes very early on, even before you exhibit symptoms, that this person is going to develop Parkinson, then, because we're not scanning brains of people in a regular basis. So you go to the doctor because you have a complaint, and by the time that they go, it's kind of already late. That's the problem, because the brain is already so affected, because it's really that specific part of the brain that dies.
Okay, and
so
the reason why it affects, the reason why you don't have loss of memory or other kind of symptoms, is it because of the specific part of the brain? Okay, so it's the specific part that has problem with the organoids, but it's not in the DNA or whatsoever. It's really like part of the cells. Like, I mean, the death of the neuron is caused by a problematic at the organoids, but what creates that at it? Like, what's the base of it?
It's can be, well, we know it's a risk if you have a mutation or a change in certain genes, but that's only about 10% of the cases, and the rest we don't know what causes Parkinson's disease.
Okay.
So there's supposedly about 90 different genes that are associated with an increased risk of getting Parkinson's disease. There's some that have been identified as higher risk than others, and it's thought actually to be a combination of genetic and environmental factors, perhaps exposure to pesticides, things like that. So it's the exact cause is not known, so the exact treatment is not known, but that's why we're looking at cells with defined genetic changes to see if at least, okay, in that type of cell, can we find a compound that could lead to a drug? Could we develop a drug that could treat that kind of Parkinson's disease?
Okay, and I was wondering, what is your latest discoveries on this? Like, right now, are you more looking at the effects, long-term effects, the speed it has, like to the speed of the symptoms they have to advance, or maybe the cause of this symptom or this yeah.
Well, since we're only looking at cells, we're only measuring how fast they can take up this protein that's associated with Parkinson's disease. The protein is called synuclein. And synuclein aggregates, like clumps of proteins, are thought to be bad for Parkinson's. And so people are studying how it gets into cells, how it gets out of cells, how if the cells can degrade this synuclein. And that's in the lab, we do assays to test like the speed, the rate, and then with having those tools in hand, then we can screen the different chemicals and see how it affects it.
Yeah, and we also have different mutations associated to Parkinson because, again, we work with genetic diseases, so we can try to understand what is in this mutated cells make them more vulnerable to the detrimental effects of synuclein, for example. If we can understand, then we can come up with some approaches that are personalized. So they would be suited for a certain type of mutation, maybe, but not another one. Maybe one of chemicals would work better in one, with people with certain mutations, but not so much for the other one. So that's the other potential for using iPSCs is personalized medicine, because we're studying the disease in your cells, in one person's cells. So we have all the genetic information there, and then we can go after medications that would work for them with their profile.
Okay.
Did you see the video on the website, on our website?
No, I haven't.
Okay.
So part of my work, or big part of my work, is to make videos about the work we do. So both for people that are researchers working with the iPSCs, but also for everyone that is interested in learning about it. So we do have social media with videos, and we do publish, we advertise our papers there too. So I'm gonna send you all these links and you can take a look. And then you can see our papers. Most of them are openly accessible, so you don't have to pay to read them. So you can, if you're interested to really read the science, you can read, or you can just watch the videos and learn more about that way, or you can do both. But I will make sure to send you after we are done talking.
Do I have screen sharing, Louisa?
Yeah, pretty sure. If not, let me know and I'll do it.
Okay. Okay, can you see that?
Yeah.
Okay.
I don't think we can hear the sound though. No, when you share, you have to say that you want to optimize for video sharing, because otherwise it doesn't share the audio. Sorry.
Okay, sorry, stop.
Do you want me to do it, or?
Sure.
Okay. I'll see if I remember how to do it, but it should be okay.
Stop. I'm very proud of this video because we all help contribute.
Yes, it's true.
To try to get the message across about what we do.
It's true. Just 1 minute.
And it had to be no longer than 2 minutes long, and we had to choose the voice and the music and the cartoon characters.
Neurological
Can you hear it?
Yep.
Yeah.
Yeah? Okay.
Neurological disorders can affect individuals at any point in their lives. Movement disorders like Parkinson's disease affect the lives of patients, their families, and our wider society. While there are many techniques to study the brain to help understand these diseases, challenges remain. For instance, autopsies and imaging techniques cannot provide the entire picture of what happens in the brain, and promising therapies keep failing. Also, due to the brain's complexity, brain cells cannot easily be accessed for research. Now, there's a new technology that allows us to study human brain cells in a dish. Here at the Neuro's Early Drug Discovery Unit, or EDDU, we generate different types of human brain cells using induced pluripotent stem cells, or iPSCs. iPSCs can be generated from the skin or blood of any individual. A stem cell can then be made into brain cells with the same genetic background and comparable features to cells in the patient's brain. Diseased cells carry mutations that can be corrected by using genome editing technology called CRISPR-Cas9. By combining CRISPR-Cas9 and iPSC technologies, we can look for differences between healthy and diseased brain cells and screen for drug candidates. With brain organoids made from iPSCs, we can now study multiple brain cell types. This brings us one step closer to better understanding the human brain and its disorders. The Neurose EDDU is an open science platform that actively promotes the sharing of our tools, methods, and knowledge to accelerate the discovery of new treatments for patients worldwide. Together, we can make a difference. Contact us to find out more.
Yes, it's pretty well done. And I saw at the end of the video, like you mentioned earlier, that basically you're sharing on all of this information. Does it have an effect on the money you get by the government to pursue these researches? What I mean is, by sharing everything, you'll be like less special. So will it impact it negatively or?
No, positively. Because the funders, like the government, they want you to share the information. A lot of them say, how will you keep it open science? And as part of the funding requirements, you're required to publish it in open access journals.
Mm-hmm.
So it's no longer researchers told to keep their research in a box. You say, you share it. If everybody knows what you're doing, we'll be able to progress faster. Like people did with developing COVID vaccines,
Mm-hmm.
because the researchers around the world shared their results and science was able to move very quickly. So open science is a very positive thing.
Yeah, it's a change of mentality. You did hint at something that was used to happen. You know, you used to be because things were competitive. You wanted to publish in the best journals, so you were kind of like encouraged to keep your data for yourself. But it's a model that doesn't really work for nowadays, and there is a lot of change in mentality. So there is actually a lot of funding coming to encourage open science.
Okay, I was asking because I have a family relative that is a researcher in the USA, and the way he was talking about it was like, for my publications, sometimes what I have to do is not give everything, and I have like to keep some of the things to make it more important afterwards, so that the publication is better and stronger, so it can get more money from the government. That's why I was wondering if it was the same, or maybe the system is a little bit different from the USA and Canada. I don't know.
I don't know. If you feel something might be better in another paper, maybe it's a matter of building a story. And sometimes if something that goes, it'll be developing a system and it'll be better quality.
Mm-hmm.
You might want to save it for your next publication. But whether it's published in an open access, I don't know.
I think for us, we're kind of in a comfortable position because what we do is kind of specialized, and we're kind of very good at doing what we're doing, developing these models, that we always have people wanting to collaborate with us. And we don't, our lab is also different. We don't have that much, it's not like a typical research lab, because we do a bit of, drug development, and we also have funding from industry and things like that. We can't afford not always just relying on public funding. So I understand that smaller labs will feel the pressure, if they have too much competition, that they can't get ahead because just the system expects them to have X, Y, or Z amount of publications. So we are kind of in a comfortable position also because there's not a lot of labs that do what we do. So, so I understand that for some people it's still a bit difficult to totally adapt to open science, but it doesn't mean that they have to do open science like from the get-go. Maybe, you know, they want to keep some of their data from being shared right away, but then once they publish, if they do open access publication, then the data is there for everyone, right? And someone can build on this data. As if you just do magazines that are not open, then people have to pay a subscription to access. So you already put one more barrier to it. So maybe when your project is still developing and you're not ready for publishing, maybe you want to keep your stuff to yourself a little bit more, just, you know, to get the chance of publishing. But then once you publish in open access, then it's there for everyone. And like I said, it's then you can build your puzzle, you know, because we have the information to build on it instead of trying everyone to do the same thing.
Okay. Great. All right. Well, that explains a bit because I was a little bit confused at first.
Yeah, it's not a model that I think there's still, I don't know if the US has the same kind of encouragement. I feel that at least here in Montreal, we are very privileged that we have the neuro, the institute, the hospital and institute where we work with works under an open science model. So every lab is doing that, you know.
I just looked it up. The NIH, which is the National Institutes of Health in the state, ensures that the public access policy ensures that public has access to the published results of NIH-funded research. So if the NIH has funded it, they have to, the scientists have to submit their final journal manuscripts to a digital archive that can be accessed by everybody.
Okay. Yeah, that's the final result from, you know, the publication, but maybe during the process they are not so willing to share because they are worried that someone will publish first, and, you know, that's can happen too, unfortunately. Not everyone is it's loyal and plays, you know, some people play dirty still.
Yeah, I guess. All right. Well, that helps a lot. And finally, I was just wondering. Right now, you're with the cells in the dish, and you're finding for you're looking with the proteins and all of that. And I was wondering, do you have multiple multiple like places that
plates.
Sorry?
Like plates or no?
No, not plates, but multiple labs or like directions for, like, do you have multiple projects for the same subject?
Lauren will know that better than me.
So I'm trying to think of any, like, say, a particular cell line. It might be used in assays in 2 dimensions. It might be used to make the 3D organoids. It just depends on the particular projects and what's going on.
And we also have different, like, we have people that are like staff, you know, they're research staff. So they might be generating the cells for someone else. But then you have the PhD and the master students that are using the cells that are being generated for their project. So one cell line can be used for multiple projects, but also we explore different diseases. So for example, we have the neurodevelopmental group, they work only with that. And then we have the group that study Parkinson, and they work basically only with that. And then we have the ALS, which is amyotrophic lateral sclerosis, the disease that Stephen Hawking had. I don't know if you know which one I'm talking about. So I think these are our three main axes. And yeah, we don't have like people, we have people collaborating, and then they might be working in the same project, but someone is leading the project and other people are supporting the project. So generating the cell lines, taking care of the cells, you know, so things like that.
And in the personalized medicine approach, you might use, you know, like several different cell lines with different mutations side by side. to compare the different cell lines with, say, one that has normal genetics.
Okay.
So on the same dish, when you've got multiple samples, you might have more than one line. It's like 4 or 5 different samples to test.
All right. Good. Well, thanks a lot. I mean, it gives me a way better understanding, and I'll read for sure the documents. I copy like the links, and yeah, thanks a lot for your time, and I appreciate it a lot.
Okay,
yes, well,
you're welcome.
Good luck with your research, and yeah, I will send you the other links to our channels, and then you can check it out if you would like.
Yeah, that would be perfect. Thanks.
Okay.
Have a great day.
Take care, you too,
Okay.
bye. Bye.
Bye.
Thank you, Lenore.
You're welcome.